Have you ever thought about how one tiny little mistake, like a single typo in a giant instruction manual, could change absolutely everything? Well, that's exactly what we're diving into today. We're going to look at what happens when the blueprint for a crucial protein in our blood has a tiny error, and we'll be using sickle cell anemia as our main example. Let's get into it. Okay, so look at these. These are red blood cells. They're basically the tireless delivery drivers for your entire body, rushing oxygen to wherever it's needed. But here's a question. What if the most important protein packed inside every single one of these cells was built from a faulty blueprint? That's not just some hypothetical question. For millions of people with sickle cell disease, this is their reality every single day. So before we can really get into what goes wrong, we first have to understand what's supposed to go right. We need to look at the basics, our blood, and the incredible powerhouse protein at the very heart of this story. We all know blood is important, but what does it really do? Well, it's kind of like the ultimate delivery service and maintenance crew all rolled into one. It carries nutrients and oxygen, hauls away waste, helps keep our body temperature just right, and even fights off diseases. It's pretty much everything to us. And now for the star of our show. Inside every single one of those red blood cells is the real hero. It's a protein that does all the heavy lifting, and it's called hemoglobin. This is the molecule that actually grabs onto oxygen when you breathe in and doesn't let go until it gets where it needs to go. And you guessed it, this is the very protein that gets messed up in sickle cell anemia. All right, so how do our bodies even make this super important protein? What's really cool is that the recipe, the genetic blueprint for it, isn't static. It actually changes as we grow up. It's like we get a software update at different stages of life. Let's take a look at how that works. At its core, a hemoglobin molecule is pretty elegant. It's made of 4 smaller protein chains. Think of it like a little structure built from 4 pieces. You've got 2 alpha chains and 2 non-alpha chains. Now, the alpha chains pretty much stay the same, but those non-alpha chains, that's where things get interesting, because our bodies swap those out depending on how old we are. And you can see that process right here. When we're just embryos, our bodies use one set of chains. Then, as a fetus, it switches to different chains called gamma chains, which are amazing at grabbing oxygen from a mother's bloodstream. But after we're born, our bodies make one last big switch to what are called beta chains. This makes our adult hemoglobin. And that beta chain, that's the one you really need to remember. It's central to everything we're talking about. This chart just breaks it down even more clearly. You can see how specific the formula is at each stage. And look at that adult version, HbA. That's the one with 2 alpha and 2 beta chains, and it makes up almost all, like 98% of the hemoglobin in an adult. It's a perfectly engineered molecule. So that brings us to the big question. What happens if the instructions to build that all-important beta chain have a mistake? Okay, this is the turning point. We've seen how the system is supposed to work, how perfect the design is. Now we're going to look at what happens when just one tiny typo gets into the genetic code for that beta chain. And that one little error, it doesn't just make a slightly off protein. Nope, it creates something completely different, what scientists call an abnormal hemoglobin. And these are molecules that just can't do their job right, which sets off a whole chain reaction of problems, the kind we see in sickle cell. And really, this quote says it all. A genetic mutation, or typo, in the gene causes an alteration in the protein. It's a direct link. It's like having one single error in a complex architectural blueprint. The foundation might be the same, but the final building ends up structurally unsound. So let's make this crystal clear by focusing on the most well-known example of this whole phenomenon. We're going to zoom right in on the specific typo that causes sickle cell anemia. And there it is. This one single genetic change in the blueprint for that beta chain creates this new, faulty version called hemoglobin S. This isn't that healthy, normal HbA we talked about before. This is the molecule that causes red blood cells to deform into that rigid sickle shape. All the painful and life-threatening complications of sickle cell anemia stem from this one protein, which stems from one typo. There's actually a name for these kinds of genetic disorders. They're called hemoglobinopathies. It's a big word, but it really just describes exactly what we've been talking about. A problem that starts with a bad gene, which creates a badly shaped hemoglobin protein, which then leads to a disease. It's a clear line of cause and effect. So, there you have it. We went from this perfectly divined molecule that keeps us alive to a single tiny typo in our DNA that can change absolutely everything. It's just a stunning reminder of how incredibly complex and delicate our biology can be. It really makes you wonder, right? If one single letter in our genetic code can have such a massive impact, what other secrets are just waiting to be read in there?